Good morning to everyone. Welcome in Trieste and welcome to ESOF 2020. I'm Simona Martinotti and now I'm a researcher in physiology at the University of Piemonte Orientale. It's not my first ESOF. My first was 10 years ago in 2010, and in 2010 I was a PhD student. So in this time something has happened, and for me it's a pleasure today is the moderator of this round table that the organizer accept when I propose it. This round table has a strange title, To Be or Not to Be, because our round table is focused on the beehive, on the honeybee word, the honeybee product, and we observe this word. Oh, Dr. Filipic, if you want to reach us on the stage.
Sit here.
And we observe this fabulous word with different type of eyes. The eyes of a naturalist, the eyes of an immunologist, and the eyes of a cell biologist, because we need to know honeybee, but we need to know how to use not only for a cake the honeybee product and the beehive product. So today are with me three speakers, Dr. Paolo Fontana, Dr. Bratko Filipic, and Dr. Elia Ranzato. And we can start with Dr. Fontana, that is a naturalist, an entomologist, and an apidologist. Until 2009, he was called by Edmund Mack Foundation in San Michele all'Adige in Trento, Italy, to form a group research on honeybee and beekeeping. He's author of 280 scientific and popular publications, and he has written several monographs, including Il piacere delle api in 2017, that in 2019 was also edited in English, The Joy of Bee. Paolo Fontana has described 98 new arthropod taxa to date, and over the years he's set up an entomological collection of over 25,000 of specimens entirely donated to the Foundation of Civic Museum of Rovereto in 2018. So, Dr. Fontana, the stage is yours. Thank you very much.
So. Okay. Good morning to everyone, to the people is connected thanks to internet. I apologize for my macaronic English. English is only my third language. The first is Veneto, the second is Italian, and the third is English, is supposed to be English. So what I will go to speak about is the relevant ecological rule of Western honeybee, Apis mellifera, because we don't really know this organism. Because when we think to honeybee, normally we think to this kind of situation. A lot of hives, well built, perfect, in line, one close to each other. Or we think to honey, the sweet honey, the wonderful honey, the food of gods. Or obviously we think to beekeeping. I am a beekeeper since 30 years, and I think that is one of the most beautiful and fascinating activity that Homo sapiens can do in his life. But even when honeybee, Apis mellifera, is not managed by humans, by beekeeping, most of people and even scientists is forced to consider it something different, something not belonging to biodiversity. There are several scientific papers that investigate this fact and and that push us to consider Apis mellifera, the Western honeybee, like an organism different from all the other species that compound biodiversity. So there is this dualism, Apis mellifera as a domesticated animal and the wild bees. And this always contribute to leave the study and the consideration of this organism to a zootechnical approach, to as a organism used only for human purposes. You can find a lot of paper in the literature about invasion of honeybees, of competition of honeybees with other bees. And the most interesting, according to me, approach is to consider a different situation where honeybees managed is introduced in the habitat. There are, honeybees is divided in more than 30 subspecies in his original distribution that you see. This yellow part of the map is the original territory where Apis mellifera was evolved. And here there are more than 30 subspecies. Here is the region where the other species of the genus Apis are present, and the Americas and Australia are places where Apis mellifera have been introduced by man for the production of honey and for the pollination of the crops. So it's different when we consider that the Western honeybee is introduced in a country, in a place where there are other subspecies of Apis mellifera, or if we introduce Apis mellifera in country where there are other species of the genus Apis, or when we introduce Apis mellifera in countries, in territories where the genus Apis is not present. But what is difficult to accept for beekeepers, sometimes for scientists and for people, is that Apis mellifera, also having been kept by humans since thousands of years, we can imagine that beekeeping started about 8,000 years ago, more or less when agriculture was adopted by humans. But in all these thousands of years, man was not able to domesticate these animals. These animals, even when managed by beekeepers, by men, still is a wild animals. And this idea of Western honeybee as a wild and not a domesticated animal is very ancient. Even Pliny the Elder wrote in his Naturalis Historiae that honeybee cannot be considered a domesticated animal, but not fully a wild animal because it's possible to keep it. But we must wait for the great Charles Robert Darwin to realize why Apis mellifera is not possible to domesticate Apis mellifera. Because this insect must find his food in the wild. You can feed the cow to obtain the milk. You can feed the chicken to obtain the meat of the chicken. But if you want to obtain the honey, bees must fly and search from their own the nectar on the or the honeydew from flowers and from plants. But the other aspect is that the mating system of these insects, it's very peculiar, and the virgin queens hatched from the colonies must fly very far from their colony to mate with drones, the male of honeybees, and this mating happened several kilometers from the hive and several meters in the high, and man cannot control this mating. If you cannot control mating of an animal, you can do a real selection and you can control the stock of your eventual selection. So what we must think clearly that Apis mellifera, the western honeybee, is part of the biodiversity of the pollinators of the group of the bees, the so-called Apoidea apiformes in scientific term. And even when managed, not only the unmanaged colonies, but even when managed, honeybee has a life history of wild animals. So beekeeping is a very strange activity because it's a productive activity, is an activity to obtain food, chemicals, pollination service, but is done managing an animal that remain totally wild in his life. So why honeybee and the other bees are so important? It's important because they evolved together the most relevant group of plants in the world that are Magnoliophytae. About 100,000 years ago, this plant started to spread in our planet, and this spread probably is, it happened because in the same period also insects start to interact with the plants, and this co-evolution made the insect very strong and the plant very efficient in their reproduction. But what is different within the different animals involved in pollination is that some animal, like this is a fly that go to flowers to suck nectar or to eat some pollen, but the larvae of this fly, it is a syrphide, feed on aphids, feed on other insects. So the life of these insects is not totally related to the plants. For the same is the colibri. The colibri is a very famous pollinator bird that sucks the nectar from the flowers. It collects some pollen and then pollinates the next flower. But when he feeds the young, he cannot offer them only nectar. He must spray insect and so on, and even do the bath. Bats that in some countries where there are flowers that bloom during the night for the purpose to allow to the bats to pollinate them, but they are predators. But the difference with the bees, with all the bees, there are about 25,000 species in the world and 1,000 in Italy, is that all their life cycle is related to the flowers because their true food is not the nectar but is pollen. And why they are so strictly related to pollen? Because they found very easily pollen for their anatomic structure. So when we think to flowers, the color of the flowers, the smell of the flowers, the shape of the flowers, they are advertisement for the organism to tell, did you find some nectar, some very nutrient pollen? Here, if you want to find other nectar and other pollen, you must go to a flower with the same color, the same shape, the same smell. But this message works absolutely very well for bees because they have the body covered by hairs that are like feathers. And by the laws of physics, electrostatic, the body of bees became charged electrostatically with a positive charge. And when they fly close to a flower, automatically, like the Swiffer, sorry for this advertisement, the bees collect automatically the pollen. The pollen is attracted by the whole body of the bees, and then they have peculiar structure, anatomical structure in the body or in the legs to put together this pollen that is the true food of the bees. So the fact that the adult and the young, the larvae, feed on pollen is what make the relation between bees and flower so strict. And this relation made a very strong walk of co-evolution between bees and flowers, and all these species evolved their own way of life. There are solitary bees, each female rear some young and new individual like this osmia, or there are this is osmia, or there are the bombus that have temporarily society. There are species with long-term society, eusociality, as in Apis mellifera. So, what is very important is this step. Bees are the best pollinators because they absolutely need to collect pollen from the flowers for their life. They found proteins, lipids, vitamins in pollen. And nectar, honeydew, is an energy source and they need for their life. Is the gasoline to fill the tank of the truck to go to collect pollen, because they feed on pollen, and so their life is absolutely related for their life. All these insects are strictly connected one to each other, and we cannot say biodiversity can survive only with bombus, only with apis, only with osmia. They must interact all together. So, can beekeeping that is responsible of the keeping of so many colonies of honeybees have an ecological role? We must consider the specific, the peculiarity of honeybee. Honeybee has a very strong animals, a superorganism that built very big colonies, and they have a lot of steps that made these species so relevant. So the wax that allow to honeybees to store for years honey, the nervous system, they have a very strong communication. They have different rules. The worker bee make different works during its life. The swarming activity, the adaptation to different climate and plants, the subdivision in subspecies too is a strategy to survive. The mating system, the polyandry, the fact that every queen of the bees mate with several males, and the rule of the drones too. So, to consider the ecological impact of honeybee, we must only do some calculation. We can have about 100,000, 200,000 worker bees, foragers bees in a colony per year. They can visit 3,000, 5,000 flowers in a day, and they can collect forage for 10, 20 days. So we can calculate the minimum and the maximum impact of a colony of honeybee in the habitat, and we realize that they can pollinate from 3 to 20 billions of flowers. It's incredible. And they can do along all the year, almost all the year, because when there are temperatures above 10 degrees, the honeybees can fly. And they can do in a very big area, because normally they fly within 1 and 3 kilometers. So from 3 to 20 billions of flowers in an area of about 30 square kilometers. So one single colony is a very extraordinary pollination machine. That's why the role in agriculture is so strong. So the interaction between honeybees and habitat and agriculture is absolutely strong. Not important if they are wild, managed with a traditional beekeeping or with modern beekeeping. Even the evolution of the bees, honeybees, and their introduction in Europe after the Ice Age tell us that the most relevant part of our flora is due to the action, the selective action of Apis mellifera. So the fact that this animal must interact with the habitat means that they can have a kind of prevision of what will happen in one habitat. Because for a colony of honeybees, it's necessary that they grow for 1-2 months more and more broad to arrive in the perfect moment when the higher amount of pollen and nectar can be collected. So this strategy, the ecological strategy of Western honeybee is to grow very big society to collect a lot of honey, but they must collect a lot of honey to make that a very big society survive. This is the reason for which beekeeping was born. Because every year a colony of Western honeybee collect all the honey it is possible. Like in next 2 years, it is not possible to collect honey. So normally, the beekeeper can collect a huge part of the honey without affecting in any way the surviving of the colony, if the climate conditions are normal. And in these years, where the climate is very crazy, colonies and especially beekeepers are having a lot of problems. So for many thousands of years, the unmanaged colonies and the managed colonies were together. There was a genetical exchange between the managed and unmanaged colonies, but in the 80s arrived this parasite, Varroa destructor. So Varroa destructor killed most of the colonies, and beekeepers start to treat their colonies, and only most of these colonies survived. The unmanaged colonies did not survive in a significant number, and this exchange between managed and unmanaged colonies stopped. So the collapse of the unmanaged colonies put in risk the conservation of the Apis mellifera biodiversity, and is a factor that makes much stronger the other problem, the pesticides, the climate change, and although we can register an increase of total number of hive managed by beekeepers, this number don't take account of the year by year loss of colonies. So beekeepers must grow every year a higher number of colonies to obtain the same or a good effect as in the past. So we don't know exactly what is the situation of the unmanaged colonies, and we don't know what was in the past, but we can only try to study right now what is the situation. We know where and how much are the colonies of Apis mellifera managed by beekeepers, but we don't know exactly how much and how they live the unmanaged colonies that are very important for the conservation of the species and even for to give a future to beekeeping. So we must to study these unmanaged colonies, and that for this reason that in Edmund Mack Foundation, in cooperation with World Biodiversity Association, we created an app to make an activity of citizen science to study the distribution and the surviving during the time of these unmanaged bees. So this app, Bee Wild, is a free app without advertisement and so on, and can be downloaded by Play Store and App Store. So, in conclusion, what is absolutely important is that Apis mellifera, the western honeybee, is an organism kept by man in beekeeping. This is very important for our health, we will see, because we obtain products extraordinary for our health, very interesting by the sensorial point of view. They are very delicious. The keeping of bees is important for agriculture, for the pollination. 1/3 of the food we eat is due to the pollination of bees and especially of the western honeybee. But we cannot forget that this animal, the same animal, Apis mellifera, is so important for the conservation of the habitat, for the conservation of biodiversity. So we must imagine a different way to manage an organism for our purpose. We must improve the productivity, but we must protect in the environment, because we cannot share, we cannot divide this organism in managed and unmanaged. It is only one species. They work in the same way, the unmanaged colonies and the managed colonies. Thank you very much.
Thank you, Dr. Fontana. So there are some questions from the audience. I have a question for you. Why do you decide to use a citizen science action to monitor and find these wild Apis mellifera and colonies of Apis mellifera?
Citizen science is a new approach that is perfect when you have a simple question and each citizen can, maybe helped with some information very simple, give a simple answer. I cannot ask to the citizens how many species of butterflies you see in your garden, because citizens are not able to divide the different species in a garden. But I can ask, do you see a colony of Apis mellifera in your garden, in the public garden, in a tree, in a wood? Because this animal is quite easy to be recognized, and because it is easy for person in charge to govern this citizen science to select the right data from the wrong data. So with some simple question, with 1 or 2 photographs, it's possible to say, yes, this data is okay. And citizen science is necessary when you want to study a phenomena in a wide way, because scientists cannot go everywhere, every time to check. So citizens can be sensible to this, and it's also, there is another goal in citizen science, not only to collect data, but to involve people in your project and to transfer sensibility to the citizens.
I think that this project will have a lot of data from citizens because citizen. Oh, yeah. Near the microphone.
At least in Slovenia, we have now developed a very relatively big number of beekeeping in towns. It means they are on tower of the house of the blocks, and that was also the reason that we studied their products, let's say bee propolis, and we tested the antimicrobial activity because we found that the antimicrobial activity of these propolis is higher than from beekeeping from
Wild.
Wild, yes.
The urban beekeeping, it's the oldest beekeeping we know, because in the past hives were not lived in the countryside, but was kept near the house or over the house. And it's very fascinating to investigate by a historical and by a scientific point of view the strict relation we have with these insects.
So I think that honeybee are really very fantastic, interesting insect. But we now need to move faster to another point of view, looking to the beehive product. And so I'm proud to present you Dr. Bratko Filipic, that is a biologist like me. But in particular, he is a microbiologist. He has a lot of field of interest: microbiology, virology, biotechnology, but also interferons, cytokines, viruses, propolis, royal jelly, and tissue culture. In particular, he extensively studied the antimicrobial and immunomodulatory activity of water-soluble and ethanol-soluble propolis of different origins: Slovenian, German, Italian, Portugal, Spain, and from Morocco. He is included into the international IHC cooperation about biophysical and microbiological examination of propolis samples of different European origins. So, Dr. Filipic, the stage is yours.
I am Bratko Filipic. I'm coming from Slovenia, and I will present the no, how it works. No, this is not the first one. No.
Sorry, there is a little problem. I think that it is the first visible one, but not all
Sorry.
Ah.
Yeah. I will present my explanation about propolis extracts enhance the antimicrobial activity of Italian and Slovenian honeys. So we have here the plan of the experiment. At first, we have two propolis extract. It was one is a water-soluble propolis grade 120 from B Natural, and second is PVS propolis extract from beekeeping Ljubljana Center. So, and that was the reason why I said that we studied the town beekeeping bee products, and these propolis extract were mixed with Italian honeys and with Slovenian honeys. From Italian honeys, we studied bioforest honey, forest honey, and flower honey. From Slovenian, we have chestnut honey, forest honey, and flower honey. I have to say that the main source of all these honeys are commercial honeys. So there is no influence of source or putting some ideas what the result should be, and so on. And so with each of these honey and water-soluble propolis extract, we prepared a mixture of composed from 90% of each of honey, and add to each of these 10% of water-soluble grade 120 and or PBS propolis extract. And this mixture were then analyzed, and we determined the minimal inhibitory concentration for different microorganisms, and determined how is grade 120 better or not better to PBS propolis extract. So at first we will explain what is the composition of propolis extract. Here you see the 100 grade 120, and you see here nice composition of different polyphenol fraction. And here you see is not very clear, but is just the information, the composition from different polyphenols. And now we go to PBS propolis extract, and you see it has only total flavonoids, caffeic acid version, chrysine, pinocembrin, and galangin. In comparison to previous one, these decided polyphenols were much, in principle, much higher amount than in grade 120. So at first we measured the antimicrobial activity of compare antimicrobial activity of grade to PBS propolis extract. And you see mainly better activity because for minimal inhibitory concentration, the lower is the value, bigger is the activity. And here at the start, PBS is better with higher antimicrobial activity to gram-positive bacteria, especially to Streptococcus agalactiae and to MRSA. MRSA is very important as human pathogen. And then we started the experiments with each of the either grade 120, either PBS extract and Slovenian honeys. So we have, in Italian honeys, sorry. We have bioforest honey at yellow, and then in second line is grade. We add grade to the honey, and here you see the red data that they are very low minimal inhibitory concentration. So it strongly increase the antimicrobial activity. But the PBS is not so good in this range. If we go, then the second is green one, forest honey. When we add the grade, you see smaller, and again bit higher the PBS extract. And next one, when we combine all in when we check which is better, we see that the grade is better for the increasing of activity, and we have here the maximum increase of gram-negative bacteria, with exception of Streptococcus pyogenes in bioforest honey. Unfortunately, bioforest honey was not so good as forest and flower honey from Italian. We don't know why, but probably is this dependent from the polyphenol composition that influence the increase or decrease. And now we have enhancing of PBS propolis extract to forest honey, to Slovenian. Forest honey, and here you see again the red one is great, is higher activity than PBS extract. And then come the biggest one, the flower honey, which has originally the weakest activity in both cases, in Italian and in Slovenia. The flower honey is not comparable to forest honey. And then we are going further. Then we have effectiveness. The Italian bioforest honey, and you see, sorry. And now next No, where are the rest of the... Oh, honey, flower honey, forest honey, Slovenian, then is flower honey, forest, flower, no, chestnut, we somehow we lost it. But now to conclude again, what I said, the highest here is increase for the gram-negative bacteria, and if the effectiveness of grade 120 was expressed is in forest honey, and with highest increase to gram-positive bacteria and Candida albicans. And when we have for Slovenian honeys, the highest increase was for chestnut honey and gram-positive bacteria. So in conclusion, it seems that the enhancement of antimicrobial activity of honey depends on sort and polyphenol composition of the honey, and not directly on PBX extract of propolis or grade 120. So in principle, effect of enhancement go by addition of polyphenol from propolis extract from B natural or PBS extract. So, thank you. Was I not?
You can. Any question from the audience? No, so why there is this bigger diversity in polyphenol concentration in the same type of honey from Italy and from Slovenia? What is the
main reason is that the eco surrounding in Slovenia is different from eco surrounding in Italy.
Oh, yeah.
Even we say we have two forest honeys, but these honeys are collected from a little bit different eco surrounding. So
we need to preserve this kind of ecosystem to maintain this.
Yeah.
So, any question? So, thank you very much. So, we move to the last speaker, that is Dr. Elia Ranzato. Is my colleague, so he's from University of Piemonte Orientale, and he is my young mentor. He is a cellular and molecular biologist. He's actually using honey as novel natural compound for wound and tissue repair and regeneration. His project is undertaken to add value to the existing honey resource by developing products with therapeutic benefits. This process involved the identification of the appropriate floral sources, the evaluation of the active agents in the honey, and the understanding of cellular and molecular events occurring during wound repair and regeneration process. So, Elia, the stage is yours.
Thank you very much, Simona. Thank you to all people present. My work is dedicated, as Simona suggested, to study the biological activity and biological properties of some honeybee product like honey, like propolis and honeydew honey. My university and my group is working in Piemonte, in Italy, in two different cities, Alessandria and Vercelli, and our research group is focused to study by different technologies, medical, natural, and bioengineered product for tissue repair. In the past, we utilized different medical products, different natural products, and different engineerable materials. Our goal is to obtain and to study biological effect of these compounds. In particular, in the past, we have utilized different natural products, such as jojoba oil, such as Ophioglossum vulgatum, a classic fern from Alps, lichens, and now, from 10 years, we are studying honey and other honeybee products. What is honey? You know, honey is a sugar solution. Honey is mainly composed of sugar such as glucose and fructose, but in honey we can find also water, around 20%, and a very small percentage of honey contain important mineral, vitamins, and other protein, mainly enzyme obtained from bees. We know from very ancient time that honey is very useful for human health. In fact, the history between humankind and bees is a very old history. In the past, humankind utilized honey not only as a food, but also as an ointment and a drug. Now, there are several products available all around the world containing honey, in particular Manuka honey, obtained from Leptosphaermum scoparium, a Manuka tree, New Zealand tree, and this product containing honey are very useful to treat wounds and other skin disorder. We know that honey contain antibacterial molecules. In particular, honey is able to produce a very strong antibacterial properties. And from several years, we know that this antibacterial activity of honey is mainly due to hydrogen peroxide production. It looks like very strange that honey contains hydrogen peroxide, but all kinds of honey produce a small quantity of hydrogen peroxide, and this hydrogen peroxide production is due to a chemical reaction due to an enzyme produced by honey sweet glands. This enzyme is called glucose oxidase and breaks down glucose to obtain glucolactone and hydrogen peroxide. So honey produces continuously hydrogen peroxide. And this hydrogen peroxide would be very useful, we have demonstrated in the past years, to induce wound repair. So we know that honey possesses antibacterial activities, so honey could be very useful to treat bacteria and yeast in the wound bed. But 10 years ago, our group asked if honey can influence the phenotype of skin cells, mainly keratinocytes and fibroblasts. We asked to ourselves if honey is able to modulate the wound closure, to modulate the collagen and other extracellular matrix proteins, and if honey is able to induce an extracellular matrix remodeling. We obtained 10 years ago a first grant from a Japanese foundation, Yamada Bee Farm Foundation, that support our groundbreaking idea to demonstrate the role of honey, different kind of honeys, in wound repair and regeneration. According to this grant, we utilized 3 different kind of honeys, representative of different kind of honey we can find everywhere all around the world. We utilize Leptospermum scoparium honey. Manuka honey is a very expensive honey coming from New Zealand. A very common honey like acacia honey obtained from Robinia pseudoacacia, and then we utilize another dark honey, buckwheat honey obtained from Phagopyrum species. We utilize an in vitro approach to demonstrate the role of honey on skin cell, and we utilize the two cell line, keratinocyte, human keratinocyte, and human fibroblast. First of all, we demonstrate the low toxicity of honey on skin cells, and we demonstrate that honey is safe to treat skin cells. But it was not enough. We utilize another very well-known test, in vitro test, to demonstrate the efficacy of honey to close the wound. We utilize this scratch wound assay. We made a scratch on a confluent cell monolayer, and we observed after honey exposure if honey was able to increase the wound closure, and in which extent honey was able to induce this wound closure. We obtained a very groundbreaking result because we demonstrate that, in particular with keratinocyte, honey was able to induce a better and a strong wound closure. We compared our result with a very classic product obtained from hospital, platelet lysate, that I have indicated as PL. PL is platelet lysate. Platelet lysate are platelets from human blood, and platelets contain a lot of growth factor, very useful to treat the wounds. So we compare the honey effect with the best product available in the hospital. Our result were very particular, very significant, because acacia, manuka, and buckwheat honey obtain a result very similar to platelet lysate. So it was a very groundbreaking observation that honey is very effective in inducing wound closure. The same result with less extent we obtain with fibroblasts, but in fact fibroblasts are the second line of cell involved in wound closure, are the keratinocyte, the cell directly involved to close the wound. Then we utilize another test to demonstrate that an important intracellular ion, calcium, is involved in wound effect upon honey exposure. We utilize confocal microscopy to demonstrate with a fluorescent probe sensitive to calcium ion oscillation that upon honey exposure, the intracellular calcium concentration are modifying. Why? To shorten this long history to demonstrate the role of honey in inducing calcium effect, we obtain, after several years of experiments, we obtained this model we have recently published. Hydrogen peroxide produced by honey in the intracellular space outside the cells can enter the cells by aquaporin 3 channel. This hydrogen peroxide entering in the cells, in the cytoplasm of skin cells, can induce in turn the opening of two calcium channels present in the plasma membrane of skin cells. In particular, TRPM2 channel and ORI1 channel. The increase of calcium in cytoplasm, in intercellular space, induce the positive effect of honey in wound closure, in keratinocyte, and then in fibroblast. These results are very interesting because we demonstrate for the first time the involvement of aquaporin 3 in the effect of honey in skin cells. In particular, we believe that this aquaporin 3 involvement is very important because we can modulate in the future by honey exposure some skin disorder, not only wounds, but other skin disorder due to aquaporin downregulation in skin, in fibroblasts and keratinocytes. So in this first part of our work, the work of my group, we demonstrate that honey is really able to facilitate wound repair process in different way. The first effect of honey in wound repair is this antiseptic action. So honey induces a reduction of growth and yeast, of bacteria and yeast growth in the wound bed. But the most important effect that we have demonstrated is that honey is able to positively influence the skin cells to grow and to repair the wounds. In particular, we demonstrate that honey is able to induce this reepithelialization phase after the initial antiseptic action. In particular, we have demonstrated that honey is able to modify the organization of cells to induce proliferation and an increase of cell motility of keratinocyte, and I have no time to describe the experiments and the data, but honey is able to induce a very important phenomenon on keratinocyte called EMT, epithelial to mesenchymal transition. This epithelial to mesenchymal transition is induced keratinocyte to proliferate and migrate to close the wound, so to allow the closure of the wound and tissue regeneration. Last but not the least, we observe also that different honey can induce different mechanisms in skin cell with different extent. So our idea is the combination honey of different floral origin can induce the best therapeutic potential and properties. But from the beehive we can obtain other very important products such as honeydew honey, in Italian miele di melata. This we test some years ago if also honeydew honey with its very different composition is able to induce the wound closure. And we utilize different kind of honeydew honey, but we observe that honeydew honey is able with less extent to induce the wound repair. So the comparison between floral honey, classic honey, and honeydew honey demonstrate that the best one is floral honey, maybe due to the production of hydrogen peroxide and maybe for presence of polyphenols, some kind of polyphenol in floral honey. Another very important product of beehive is the propolis. It's very strange history of propolis, as Dr. Brakto Filipic described a few minutes ago. And propolis may be a new frontiers for wound healing. We published with Simona this review 5 years ago on burns and trauma. It's very strange. It was a very small review with a few data available in the scientific literature, but this paper from 5 years is one of the best 5 cited paper of burns and trauma. So it looked like that bee products, in particular propolis, are very interesting for other scientists to treat wound repair, to induce wound repair. We utilize the classic scratch wound assay to demonstrate if propolis is able to influence in a positive manner the behavior of keratinocyte, and in fact we demonstrate that propolis is better than floral honey to induce the wound closure. You can see the comparison with the platelet lysate, the same growth factor of platelets, of human platelets, that some hospitals, also in Italy, are still utilizing to treat difficult to heal wounds. And if you can see that the propolis at 0.001% can induce in vitro model wound repair in a better way than platelet lysate. It was a very groundbreaking result. Propolis is a very effective in wound repair. Which is the mechanism of propolis? It seems very strange, but also propolis induce inside the production of a low amount of hydrogen peroxide. So it seems the same history of honey. Aquaporin tree is involved again in keratinocyte and fibroblast to allow this hydrogen peroxide produced by propolis outside the cell to enter the cells. In fact, we demonstrate that aquaporin 3 is involved in this mechanism. We observe an increase of the gene of a protein, aquaporin 3, in keratinocyte exposed to propolis. So the history look like very similar. The hydrogen peroxide produced by honey and propolis entered the cells by aquaporin 3 channel in the plasma membrane. This aquaporin, this hydrogen peroxide modulate the positive effect. So taken together, we have some data that honey and honey bee products in general, such as honeydew honey and propolis, can positively influence the biology of skin and skin disorder. In particular, we demonstrate that honey and propolis positively modulate from epithelialization, so the closure of the wound. Propolis and honey modulates the granulation tissue formation. Honey and propolis increase wound contraction. Propolis and honey can induce a production of collagen, a different kind of collagen, so the modulation of extracellular matrix, very important to avoid the formation of scar after wound. And we are studying in this period the possibility of honey and propolis to modulate the behavior of endothelial cells. Endothelial cells are very important because they represent the most important tissue to deliver blood and other components of blood into the wounds. So if we can modulate, our idea is to modulate the endothelial cell regeneration utilizing honey. Of course, I present in these few minutes the 10 years of work, but it was not only my work, it was a work of our group at the University of Piemonte Orientale. This work allows us to have some important awards. Simona 7 years ago won a very important international prize, the last prize for the utilization of in vitro testing to study skin disorder and to study skin problems. And we have a lot of students and PhD students interested to characterize the effects of some natural compound, in particular honeybee product, in terms of cellular and molecular behavior of skin cells. And last but not the least, if you want, you can read this book that we published a few years ago about the cellular and molecular mechanism of honey wound healing. My take-home message is that 2 tablespoons of honey would fool a honeybee flying once around the world. So take care of bees. They are very important for our planet. They are very important also for our health. Thank you again for your attention.
Thank you very much. There are some questions from the audience. Oh, sorry. I arrive. Okay.
Yes.
Hi. What about all the other tissues that contains fibroblasts and keratinocytes? I mean, is it only related to skin cells, or we can talk about joints, for example, or bones?
Thank you for your question. It's a very interesting question. We believe that cells like keratinocyte and deriving from ectoderm, so from the external tissue of the body during the embryonic development, can be modulated by honey. We tested in the past honey on osteoblast bone cells regeneration. We didn't publish yet this result, but I think it's very interesting because we believe that this mechanism, the hydrogen peroxide production by honey and the entering of hydrogen peroxide into the cells seems to be the same mechanism. The difference between keratinocyte and other cells could be on the calcium channel involved, because TRPM2 is a typical channel for keratinocyte. I think we cannot find, or TRPM2 is not the best channel for osteoblast bone cells. So I think we need to better define the channel, the plasma membrane channel involved in hydrogen peroxide phenomena. Thank you.
Other question?
Can I?
Yeah?
Compliment for the research and the presentation. And I cooperate in some way with some friends that are involved in what is considered apitherapy. And, but the problem is that the medical community, especially in Italy, is still reluctant to approach these products, probably because there are not so strong economical interest right now. But what do you think about the growing of the medical community on this aspect?
Well, I think there are too many problems utilizing the honey for as medical product or product for other medical application. The first problem is which kind of honey we can utilize, and the second one is the reluctance of medical community to use these strange natural products. For the first problem, which kind of honey we can utilize, at the moment the best one that is available for wound repair is Manuka honey, obtained from Leptospermum scoparium New Zealand tree. It's a very expensive honey because you can buy a bottle of Manuka honey for around 40-50 euros. So there is a strong economical problem about the use of Manuka honey. Our data suggests that there are other dark honey with the same or better result of Manuka honey. So we can rethink about which kind of honey we can utilize. In general, the darkest is the honey, the stronger is the effect on wound repair. For the second problem, I think the work of our group, the work of Simona, is very important to put the basis of cellular and molecular characterization. In the past, data available from honey utilization wounds were only anecdotal data. We utilize honey, we obtain very interesting result. Now I think with the characterization of cellular mechanism, molecular events involved in skin regeneration upon honey exposure can boost the use of honey. And if you consider honey is also very important because utilizing honey you can influence the skin regeneration, but you can also influence the wound bed, avoiding the growth of bacteria and yeast. So with the same product we obtain two different results, two very effective results. For the antibacterial activity, the hydrogen peroxide seems to be the most important effect of honey, the most important activity, but there are also data that we have published some years ago with a group from Slovak Academy of Science that there are some small peptide, small protein like defensin that can cooperate with hydrogen peroxide to avoid the growth of bacteria in the wound area.
There is a question from the web.
Nice.
Which are the skin diseases in which aquaporin misregulation is involved?
The best one that I know is psoriasis, but I think in general the problem about the skin hydration, the water, the passage of water across the skin keratinocyte are different kind of disorder that rely on dysregulation of aquaporin expression. So the use of honeybee products for wound repair boost. Some question from the audience.
Okay. Thank you for your talk. It was very nice.
Thank you.
If you use it for medical applications, so would you just like spread it in a wound, or how would you use it, the honey?
Very good question. Thank you. I don't know exactly. I think it's the most important question about the use of honey is which is the best way for honey application. It depends on the type of wounds. If you are wounds open to the environment, I believe the best way is to use, I don't remember the English for una benda in English, bandage with wounds. In fact, with honey, in fact, there are some products also available in Italy, very expensive, bandage containing honey to treat exposed wounds. For small wounds without direct exposition to the hair, I think also the treatment directly with honey could be very useful. I have also some data about which is the concentration, the best concentration for honey, and I think we utilize in the in vitro experiment concentration of honey around 0.51% of honey. I think the same concentration are very easy to reach with this product containing Manuka honey at the moment. On the market, you can find at the moment only product containing Manuka honey. I believe in the future that other medical product can contain not only Manuka honey, but also other kind of honey, because we demonstrate that not only Manuka is very effective in inducing wound repair, but also other honey can represent a new market for apidologists.
If I can, I think that in the future with new technology like nanotechnology or something like this, we can put this type of product directly also during surgery in the safe way. So is the challenge for the next year, for the future. So
for propolis, the problem I think is quite different because we utilize only hydroalcoholic propolis solution. So I think in the future we can try to utilize it, to compare, like Dr. Bracco Filippici demonstrate, other preparation of propolis, because I think it's a very interesting to compare not only hydroalcoholic preparation, but also PBS preparation or water extract of propolis. Propolis are very strange products. I think it's quite difficult to have similar product in different laboratories. So I think the challenge is different for propolis.
she has another.
Yeah.
So you showed many awards that you just work in vitro. Do you also have plans to work in vivo, or do you do all your work in vitro?
Simona, do you want to?
For now, it's only in vitro because is the type of work that we like to do. Obviously, for a clinical use of this product, we need an in vivo study, but for the screening and the characterization, we want to use only in vitro study, like in the European community is mandatory for cosmetics, for example.
I think that these in vitro studies are extremely interesting and relevant because for honey we have a very long history of use for skin, wound, burn, and I did it on my face too, and I can tell it works. It worked on me, but these studies can give a scientific on a traditional use. So probably for the main, the most common use, we don't need a lot of tests in vivo, but this in vitro can explain a lot. I would like to add another problem of the use of honey for this purpose. That is not only the way to applicate, but is the origin of honey.
Yeah.
Now we have a very big problem in the world about honey. There is false honey, honey that are introduced from a country to another in a legal or illegal way. But as I told in my presentation, bees collect in the wild. So in the environment. So in the honey, we can find a lot of good, but also ugly things. And I work in Edmund Mark Foundation since 10 years, doing environmental monitoring about with bees of the habitats. And the most important thing will be to give also some direction to the beekeepers where and how to produce honey. In my opinion, we should extract honey for medical purposes from natural combs and not from wax foundations, and with a beekeeping specifically done for this purpose. So not only different honey has different composition, but we must imagine a beekeeping
Of quality.
Of quality, yes.
Yes, I think it's a big problem because in honey, like Dr. Paolo Fontana suggests, that we can find pesticide, antibiotics, of organic, of inorganic pollutants, so it's very important, the quality of honey. In particular for Manuka honey, we know that there are a problem of quality of Manuka honey, and the price and the sophistication of this kind of honey that is very interesting for medical application.
So, other question? So I think that we can close this round table. I hope that now you consider in a different way honey, in a different way honeybee, and you spread this information and this fundamental presentation of our speaker to protect honeybee and all the product that they can produce for there, but also for our quality of life and
regeneration of our tissue if we need. So thank you very much to attend our roundtable. Thank you of all the staff that allow us this roundtable, and have a good day.